Good morning, my dear students. So, welcome to my classes. Today we are going to discuss about the topic called as RNA interference technology, which is the most important technology for your examination point of view. And if you can understand the significance of this RNA interference technology, it will be extremely useful for multiple areas. For understanding the protein synthesis, for understanding the concept called as mRNA vaccine, and it is the topic that can be useful in multiple ways. So let us start our discussion. And what is RNA interference technology? RNA interference technology. So actually, RNA interference is nothing but the RNA is the messenger that carries information from the nucleus to the ribosomes. In a cell, the nucleus is there. Inside the nucleus, you can see the presence of chromosomes. So the genetic information from the chromosomes will be copied from the chromosomes to the ribosomes. And this process of copying the genetic information from the nucleus to the ribosomes is very crucial and very important. And it is the place, okay, where the protein synthesis will occur. Okay, for copying the genetic information from the nucleus to the ribosome. Okay, so if while copying the genetic information, if any mistake happens, those mistakes has to be overcome. Okay, so those mistakes can be corrected by the technology called as RNA interference technology. So for everything, you can see, I would like to give you a brief information, brief understanding about this. If DNA is there, okay, from the DNA, we can see the conversion of DNA into mRNA, and this the mutations in the any of the DNA, those mutations will be carried by the mRNA. So the DNA, from the DNA, the genetic information will be copied to the ribosomes from the concept called as mRNA, and that mRNA, okay, that mRNA is the most important part. So this mRNA, so if any mistakes happen in DNA, those mistakes can be carried forward by the forward to the mRNA. So those mistakes has to be corrected through the insertion of another RNA, and that RNA will be entered and disrupts the entire mistakes, and those mistakes will be deleted or removed through the concept called as RNA interference technology. Our genome operates by sending instructions for the manufacture of proteins from DNA in the nucleus of the cell to the protein synthesis machinery in the cytoplasm. So what is genome? The complete structure of the DNA is called as genome. The whole functional part of the DNA is called as genome. Genome operates by sending instructions for the manufacture of proteins. Instructions for the manufacture of proteins from DNA in the nucleus of the cell to the protein synthesizing machinery in the cytoplasm. To the protein synthesizing machinery in the cytoplasm. So this transfer is the most important crucial mechanism. Transfer of the genetic information from the nucleus to the ribosome is one of the crucial player. So here in this image, you can see a clear-cut understanding of this particular area. So here, so this is the process called as protein synthesis. DNA will be converted into, so where is, this is the nucleus. Inside the nucleus, DNA is there. Through the process called as transcription, the genetic information present in the DNA, will be copied into mRNA, and now the mRNA will come out of the nucleus and enter into the cytoplasm, and so this mRNA, depending upon the genetic information copied by the mRNA, so in between the two ribosomes, there is a process called as translation. Okay, so if you understand, if you observe this very clearly, so the mistake, if any mutations happen in DNA, those mutations can be seen in mRNA. Or while copying the genetic information, if there is no mistake in DNA, so sometimes while copying the genetic information from DNA to mRNA, so there also you can see, you can witness the important concept on the mutations can be possible. So to overcome these problems, one of the most exciting technology is nothing but RNA interference technology. One of the most advanced technology is nothing but RNA interference technology. And this RNA interference technology. Okay, so this is the important. So while coming the mRNA from the nucleus to the ribosomes, from the nucleus to the ribosomes, we can see the most dramatic event is nothing but this is called as conversion of DNA into mRNA is called as transcription, and the conversion of copying the proteins depending upon the mRNA to ribosomes is called as translation. So our genome operates by sending information from double-stranded DNA in the nucleus via single-stranded mRNA. So DNA is a double helical structure, whereas mRNA is a single-stranded, to guide the synthesis of proteins in the cytoplasm. What is actually meant by this RNA interference technology? So the experiment, RNA carrying the code for a muscle protein. So how this particular RNA interference technology has been started or invented. So RNA carrying the code for a muscle protein is injected into the worm C, C elegans. So RNA coding for a muscle protein has been injected into a worm called as elegans. So this single-stranded RNA has no effect. So for example, this is sense RNA, and RNA has no effect. But when the double-stranded RNA, so one single-stranded RNA has been inserted, and that single-stranded RNA has had not shown any impact on the targeted portion, and then we cannot we cannot see any effect on it. Okay, so but when double-stranded RNA is injected, the worm starts twitching in similar way to worms carrying the defective genes. So they have chosen a insect which is having a muscle defect. Okay, so which cannot able to make any certain movements. So for making to make such movements, they have identified a protein, they have identified a RNA, and by inserting this one single-stranded RNA have not shown any impact, and they have made this double-stranded RNA, and this, because of the impact of this dsRNA or double-stranded RNA, the twitching movement has been started. So the worm starts twitching movement in a similar way to worms carrying a defective gene for the muscle protein. Means, so RNA interference technology. So by the insertion of this RNA, and that insertion, okay, so that insertion has changed the mechanism of this particular RNA protein synthesis. How it had shown. Okay, so the RNA interference mechanism. RNA interference is an important biological mechanism in the regulation of the gene expression. Okay, so it is not trying to block the genetic information. It is not trying to eliminate the genetic information. Only to in the regulation of gene expression. My dear friends, try to understand. So the words are very, very important. So you need to appropriately understand the inherent meaning of the words. So RNA interference is an important biological mechanism in the regulation of gene expression. Double-stranded RNA, so this is dsRNA, double-stranded RNA, binds to the protein dicer. So it will attach to the protein dicer, which cleaves dsRNA into smaller fragments. Okay, so dsRNA has inserted, and so where to which it has been attached to the double-stranded protein dicer. Okay, so this dicer has cleaved the dsRNA, double-stranded RNA, into two single strands. Okay, one of the RNA strands is loaded into a RISC complex. So now this has been a single-stranded RNA has been attached with the RISC complex and links the complex to the mRNA strand by base pairing. So and it links, it will be so mRNA is coming. So try to understand. So when mRNA is coming, so that mRNA which is supposed to make the proteins. So to that you can see the attachment of RISC. Okay, so you can see the attachment of this dsRNA. So one strand of the dsRNA will be attached to that. So mRNA is cleaved and destroyed. No protein can be synthesized. As no protein has been synthesized, thereby we can see the elimination of the entire unwanted features. So that is how. So let me repeat. Number one, dsRNA has inserted, and dsRNA has been attached to the protein dicer, and dsRNA after attaching to the protein dicer, and you can see the breakdown of the dsRNA, double stranded RNA, into single strands. And so this single stranded RNA molecule has been attached to the RISC complex, RISC, and this RISC complex, through the RISC complex, mRNA will be passed. And while passing the mRNA through the RISC complex, you can see the formation of mRNA will be cleaved, so at the designated location. So wherever the protein synthesis is not needed, wherever the protein synthesis is not at all needed, there you can see the elimination of the proteins. So mRNA is cleaved and destroyed. No protein can be synthesized. Okay, and several processes in the cell using RNA interference. So that is first. You need to explain the concept called as what is the central dogma, and try to draw this diagram. After that, you can go for the concept called as experiment, sense RNA, antisense RNA, and double stranded RNA. Third, you can see the RNA interference mechanism. Number four, several processes in the cell use RNA interference. When an RNA virus infects the cell, it injects its genome consisting of double stranded RNA. RNA interference destroys the viral RNA, preventing the formation of new viruses. RNA interference technology destroys the viral RNA. It will destroy what? Viral RNA, preventing the formation of new viruses. Okay, it will destroy the viral RNA. So the replication process of the viruses can also be destroyed by the RNA interference technology. Synthesis of many proteins is controlled by genes encoding microRNA. After processing microRNA, prevents the translation of mRNA to protein. So likewise, so viral RNAs will be eliminated, and viral mRNAs can also be destroyed. So first, you can see viral RNA can be eliminated. Next, microRNA. So microRNA can also be controlled. In the research laboratory, the double-stranded RNA molecules are tailor-made to activate the RISC complex to degrade mRNA for a specific gene. So that is how you can see the concept of RNA interference technology. Okay, and so try to draw this diagram. So for example, in UPSC mains, for example, if it is asked for 12.5 marks, you can try to draw the diagrams through which you can able to impress the examiner on a large line. These instructions are conveyed by messenger RNA. So the DNA information, the information present in the DNA are conveyed by the messenger RNA or mRNA. In 1998, the American scientists Andrew Fire and Craig Mello published their discovery of a mechanism that can degrade mRNA from a specific gene. The degradation of mRNA can be possible, and the degradation of mRNA at a specific gene, okay, from a specific gene can be degraded, and it has been identified by the two great scientists called as Andrew Fire and Craig Mello. Andrew Fire and Craig Mello are the two scientists in 1998 who published the discovery of a mechanism that can degrade mRNA from a specific gene. This mechanism is called as RNA interference, is activated when RNA molecules occur as double-stranded. So, if the RNA molecule is single-stranded, it is impossible for the RNA to break the entire mRNA sequence at the designated location. It is the best place whenever the RNA are you getting my point? Whenever the RNA interference, okay, RNA interference can be possible only through the dsRNA. So I hope you had seen in the image itself. So single-stranded RNA, while launching single-stranded RNA, so single-stranded RNA was unable to break that particular specific portion of the mRNA. Double-stranded RNAs are much suitable for breaking this particular area. So occur as double-stranded pairs in the cell. Double-stranded RNA activates biochemical machinery, which degrades those mRNA molecules. So only double-stranded RNA molecules will activate the biochemical processes which degrades the mRNA molecules that carry a genetic code identical to that of the double-stranded RNA. When such mRNA molecules disappear. So now if the mRNA is not there, so if the mRNA molecules gets disappeared, the corresponding gene is silenced. So isn't it or not? If the mRNA is there, so its related proteins will be produced, and it means what? The gene is produced. Okay. So we can say mRNA has been blocked. If the mRNA has been blocked, thereby we can see the silencing of the genes are possible, and no protein of the encoded type is made. And no protein of the encoded type is made. RNA interference occurs in plants, animals, and humans. It is of great importance for the regulation of gene expression, participates in defense against viral infections, and keeps jumping genes under control. So these are the broader areas of application with reference to the RNA interference technology. RNA interference occurs in plants, animals, and humans. It is of great importance for the regulation of gene expression. It is of great importance for the regulation of gene expression. And it participates defense against viral infections also. So if any invading virus is entering, so that to fight against the invading viral infection also, this RNA interference technology will play a very crucial role and keeps jumping genes under control. For example, so some genes will be used to be controlled. Here it is to be expressed. So instead of here, sometimes it may be expressed here. Okay. So if it is supposed to express at point A, and if it is expressing at point B, so instead of A to B, B to C, it is constantly jumping. You can block the jumping genes, okay, which are nothing but the transposons. Barbara McClintock's proposed transposons. Okay, you can also control jumping genes. You can keep jumping genes under control. Next, RNA interference is already being widely used in basic science as a method to study the function of genes, and it may lead to novel therapies in the future. RNA interference is already being widely used in basic science. It is widely used in basic science as a method to study the function of genes. Okay, so to observe the function of the genes, okay, to study the function of the genes, and it may lead to novel therapies in the future. So as of now, so exportation has been blocked. Exportation of the mRNA has been blocked. So if one particular activity has been stopped, means, so we can say this exportation is responsible for so and so function. If Y portion of the gene has been blocked, so Y portion of the some other function may be blocked. So a method to study the function of genes, and it may lead to novel therapies in the future. So any unwanted outcomes are there. Those unwanted outcomes can be completely controlled by this mechanism, the function of genes, and it may lead to novel therapies in the future. The flow of information in the cell. So this is the very important dynamic area. So this gives you so much conceptual clarity with reference to the entire area. DNA formation of flow of the DNA via mRNA to protein. DNA first will be converted into mRNA, and this mRNA will produces the proteins. The genetic code in DNA determines, so which type of protein has to be produced will be decided by whom? DNA. So the genetic code in DNA determines how proteins are built. So one type of DNA will able to produce one type of the same kind of proteins, and one kind of things. So will able to produce one sort. The instructions contained in the DNA are copied to mRNA and subsequently used to synthesize protein. The instructions contained in the DNA are copied. So you are directly DNA cannot able to produce the proteins. How the DNA can able to produce the proteins? Through the concept called as what? mRNA or messenger RNA. So DNA to mRNA, mRNA to protein. So we cannot alter the DNA. We can able to alter the mRNA. So altering mRNA cannot be possible by single standard RNA. So blocking the mRNA through double standard RNA is called as what? RNA interference technology. Is called as RNA interference technology. This flow of genetic information from DNA via mRNA to protein has been termed the central dogma of molecular biology by the British Nobel laureate called as Francis Crick. So Watson and Crick are the two scientists are there who has been identified the double helical structure of the protein. This flow of genetic information from DNA via mRNA to protein has been termed the central dogma of molecular biology by the British Nobel laureate Francis Crick. Proteins are involved in all processes of life. For instance, so we this can be the expected area in your preliminary or objective sort of question. Proteins are involved in all processes of life. For instance, as enzymes. Enzymes are made up of what? Proteins. Some of the hormones are made up of proteins, and some of the antibodies, interferons, cytokines, okay, interleukins. So these are all nothing but the proteins. So proteins are involved in all processes of life. For instance, as enzymes, digesting our food, receptors receiving signal in the brain, and as an antibodies defending us against the bacteria or pathogens. So proteins are involved in all processes of life. So if we can able to block the proteins, okay. So if you want to have the strong antibodies, that you can able to activate certain amount of mRNA. So if a person is supposed to have some sort of changes, okay, so some mutations, those mutations can also be blocked by this particular mRNA, by blocking mRNA. Our genome consists of approximately 30,000 genes. Our genome consists of what? No? Approximately 30,000 genes. However, only a fraction of them are used in each cell. So even though out of 30,000 genes, only a fraction of them are used in each cell. Only a fraction of them are used in each cell. Which genes are expressed is controlled by the machinery that copies DNA to mRNA. So which genes has to be expressed? Which genes are supposed to be produced? And which genes are supposed to be produced? The mRNA will be regulated by the mechanism by the mechanism called as transcription. While copying from DNA to mRNA, that process is called as transcription. That process is called as what? Transcription. Our genome consists of approximately 30,000 genes. However, only a fraction of them are used in each cell. Which genes are expressed is controlled by the machinery that copies DNA to mRNA in a process called as transcription. It in turn can be modulated by various factors. So, for example, the mRNA, regulation of the mRNA can be modulated by various factors. The fundamental principles for the regulation of gene expression were identified more than 40 years ago by the French Nobel laureate Jacob and Monod. French Nobel laureates called as Jacob Monod. So, which gene is gene protein, DNA to mRNA, mRNA to protein. So DNA, the functional part of the DNA is nothing but gene. So gene can be modulated by various factors, and the fundamental principles of the regulation of gene expression were identified more than 40 years ago by the French Nobel laureates Jacob and Monod. That is called as one gene, one protein theory. One gene, one protein theory. Today we know that similar principles operate throughout the evolution, from bacteria to humans. Every living creature is having such a tremendous mechanism, and how this particular evolution is happening, okay, throughout evolution, from bacteria to humans. They also form the basis of gene technology, in which a DNA sequence is introduced into a cell to produce new protein. For example, if you insert a new DNA sequence like Bt cotton or Bacillus thuringiensis cotton, in which a DNA sequence is introduced into a cell to produce new protein. Andrew Fire and Craig Mello were investigating how gene expression is regulated in the nematode worm Synorbatitis elegans. Injecting mRNA molecules encoding a muscle protein led to no change in the behavior of the worms. Why? So I told you we discussed while discussing the image. So why? Because one single mRNA molecule cannot able to control the mRNA. Okay, injecting mRNA molecules encoding a muscle protein led to no change in the behavior of the worms. A genetic code in mRNA is described as the sense sequence, and the injecting antisense RNA, which can pair with the mRNA, also had no effect. So first they have inserted a sense RNA, and second they have inserted antisense RNA. So for example, if you look at the GIP now, so left side if it is sense and right side it is called as antisense. So both have joined together, we can say the formation of a complete RNA, dsRNA. So first they have launched one single standard sense RNA, no effect, and they have simultaneously launched antisense RNA, even then no effect. By launching sense and antisense RNA collectively, and the effect started exhibiting, also had no effect. But when Fire and Mello injected sense and antisense RNAs together, they observed the worms displayed peculiar twitching movements. So for RNA interference technology cannot be controlled only by the one single standard RNA. dsRNA is a very much mandatory thing. Similar movements were seen in worms that completely lacked a functioning gene for the muscle protein. Similar movements were seen in worms that completely lacked a functioning gene for the muscle protein. When sense and antisense molecules meet, they bind to each other and form double-stranded RNA. And form what? Double-stranded RNA. And this double-stranded RNA, could it be that is a double-stranded RNA molecule silences the gene carrying the same code as the particular RNA? Fire and Mello tested this hypothesis by injecting double-stranded RNA molecule, containing the genetic codes for several other warm proteins, In every experiment, injection of double-stranded RNA carrying a genetic code led to silencing of the genes. In every experiment, injection of double-stranded RNA. Carrying a genetic code led to them. Okay, so double-stranded, every experiment. So Craig and Mello has proved that they have identified that. So injection of double-stranded RNA carrying a genetic code led to the silencing of the gene containing that particular gene code. The protein encoded by that gene was no longer formed. So this double-stranded RNA has blocked the specific portion of mRNA. As it has blocked the specific portion of the mRNA, the entire mRNA has been stitched together. Okay, so mRNA has been sliced, and the protein has not been formed. Okay, so the insertion of the double-stranded RNA has led to the blockage of the specific portion of the DNA. So as the specific portion of the DNA has been blocked, thereby you can see the unwanted, undesired protein has not been produced. The protein encoded by that gene was no longer formed. Also, after a series of samples but elegant experiments, Fire and Mello deduced that double-stranded RNA can silence genes. This RNA can do what? Can silence genes. That this RNA interference is specific for the gene whose code matches that of the injected RNA molecules, and that RNA interference can spread between cells and even be inherited. That is a very important thing. RNA interference can spread between the cells. So, for example, if the RNA interference has been stopped, okay, if the RNA interference has been stopped, okay, so RNA interference has happened to one particular cell. So RNA interference can spread between the cells and even be inherited. It can even be inherited. It was enough to inject tiny amounts of double-stranded RNA, to achieve an effect. So it was enough to even smaller a traces volume of double-stranded RNA has been inserted. So through the insertion of the double-stranded RNA molecule, so that we can able to expect the positive effects. Okay, so Fire and Mello therefore proposed that RNA interference is a catalytic process. What is the meaning of catalytic process? So if one small amount of double-stranded RNA has been inserted, and that small volume of dsRNA will go and control other cells, and that these cells will go and influence other cells. Likewise, catalytic, like chain reaction. So RNA interference can influence the neighboring cells. RNA interference can be inherited, and RNA interference can also be considered as a catalytic process. Double-stranded RNA binds to a protein complex, dicer, which cleaves the dicer into two fragments, which cleaves the DS double-stranded RNA molecule into two segments. Another protein RISC binds these fragments. So dsRNA, after attaching with the dicer, will be separated into two segments, and one segment will be attached to the RISC. These fragments, one of the RNA strands is eliminated. But the other remains bound to the RISC complex and serves as a probe to detect mRNA molecule. Okay, so dsRNA plus dicer. Okay, after attachment, you can see the dsRNA will be split into two single-stranded components. So this dsRNA, after converted into two single-stranded components, you can see the formation of this two single-stranded components. Among these two single-stranded components, you can see the formation of, this single-stranded component will be attached to the RISC complex. Now, single-stranded RNA and RISC complex are eligible to identify the mRNA molecule. Are eligible to identify what? mRNA molecule. When a mRNA molecule can pair with the RNA fragment of RISC, it is bound the RISC complex, cleaved and degraded. The gene served by this particular mRNA has been silenced. So once the entire mRNA, which has been attached with the after RISC plus single standard, okay, RISC plus single standard. So if it will be through this, you can see, you can expect the passage of mRNA, and mRNA will be unwanted portion of the mRNA will be blocked, and there the mRNA will be split into two fragments. Then the gene served by this mRNA has been silenced. So the gene served by this particular mRNA has been silenced. So RNA interference, it defends against viruses and jumping genes. RNA interference, it is a defense against viruses and jumping genes. RNA interference is important in the defense against viruses. RNA interference is important in the defense against viruses, particularly in lower organisms. So it is not a newly, exclusively innovated or creative thing. It is important in the defense against viruses, particularly in lower organisms. Many viruses have a genetic code that contains double stranded RNA. When such virus infects a cell, it injects its RNA molecule, which immediately binds to dicer. So many viruses. So if the virus, to block the viruses, okay, RNA is an important defense against the viruses. Many organisms, lower organisms, are having this mechanism. I personally believe this immunological, like either in the case of CRISPR technology or RNA interference technology, these two technologies are not invented things. These are already there in the nature. CRISPR is a defense mechanism of bacteria to fight against the virus. Even in the case of RNA interference is there in lower organisms. Many viruses have a genetic code that contains double-stranded RNA. When such a virus infects a cell, it injects its RNA molecule, which immediately binds to dicer. The RISC complex is activated. Okay, the viral RNA is degraded, and cell survives the infection. In addition to this defense, higher organisms such as man have developed an efficient immune defense involving antibiotics, killer cells, and interferons. Okay, to this defense, higher organisms like human beings so have developed an efficient immune system like antibodies, natural killer cells, and interferons. Jumping genes, also known as transposons. So this is also one of the important area in your examination point of view, especially for your objective related area. Jumping genes, also known as transposons, the DNA sequence that can move around the gene. They are present in all organisms and can cause damage if they end up in the wrong place. So wrong parking. So if you the gene, the DNA at the genome must be there in its original habit. If it has been moved from one place to another place, they are present in all organisms and can cause damage if they end up in the wrong place. Many transposons, jumping genes are called as what? Transposons. Technically, jumping genes are called as transposons. Generally, we call them as jumping genes, general name. Technically, we call them as transposons. Many transposons operate by copying their DNA to RNA, which is then reverse transcribed back to DNA and inserted at another site in the genome. Part of this RNA molecule is often double-stranded and can be targeted by RNA interference. In this way, RNA interference protects the genome. Okay, so wherever the through the reverse, okay, so DNA to RNA, which is then reverse transcribed back to DNA and then inserted at another site of the gene. So actually, if it is supposed to be located at A and by mistake it jumped to B, so by through the reverse transcription, so reverse transcribed back and the DNA, that portion of the gene will be again keep it at A. RNA interference regulates gene expression. RNA interference is used to regulate gene expression in the cells of humans as well as worms. It is used to regulate gene expression in the cells of humans as well as worms. Hundreds of genes in our genome encode small RNA molecules called as microRNA. Gene in our genome encodes small RNA molecules, and those small microRNA molecules will also be blocked. They contain pieces of codes of other genes. Such a microRNA molecule can form double-stranded structure and activate the RNA interference machinery to block protein synthesis. The expression of that particular gene is silenced. The expression of that particular gene is silenced. We now understand that genetic regulation by microRNA play an important role in the development of the organism and the control of cellular function. So microRNA is also one of the important molecules that can form double-stranded structures and activate the RNA interference machinery to block the protein synthesis. The expression of that particular gene is silenced. Okay, microRNAs can also be used to block the protein synthesis, and these microRNAs can able to form into double-stranded RNAs. We now understand that genetic regulation by microRNAs plays an important role in the development of the organisms and control of cellular function. Okay, so I hope you understand the concept of RNA interference technology. So you just constantly revise this topic. So through this, you can make a comprehensive information. Okay, I hope you understand. Make a notes and what is RNA interference technology. Don't never forget to draw the diagrams through which you can able to score maximum marks. In UPSC, 10 out of 12.5 marks are possible. In APPSC, 10 out of 10 marks can also be possible, particularly from the concept called as RNA interference technology. So that's it for today. Thank you very much. We'll continue in another session.